Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts in biosciences. Today we are going to discuss about the crassolucian acid metabolism, which is very commonly known as the CAM pathway. This is a pathway typically observed in plants that are growing in very arid area. There is a very interesting story how this pathway was initially identified. There was a very famous gentleman, a British gentleman who was in Delhi at that time. One day when he was examining the plants, the ornamental plants that he had in his garden, he noticed that a typical bryophyllum plant, an ornamental plant, that was tasting bitter, sour in the morning. And the same leaves he tasted in the afternoon, and he observed the sour taste has disappeared and it has become tasting more like a herb. He was very curious. When he went back to London, he reported this particular observation to the Linnean Society. We know that there are many plants that survive in very arid conditions. For example, in the cactus that is growing in the Arizona region in the United States, which is highly arid, there is very less water available, and these cactuses which are growing there are a kind of a beauty, and people have been interested in knowing how a plant can survive in such arid condition when the water level is very low. So this kind of investigations has resulted into the discovery of the crassolucian acid metabolism. When we looked at the C4 pathway, we have observed that C4 pathway has evolved by carrying out carbon fixation in two different locations, whereas in the CAM pathway has another adaptation. If the CO2 fixation will occur not in two different locations but at two different times. One will occur at daytime, another reaction in the night time. We are going to look at how this is happening. If you look at in the stomata or the leaf arrangement in the let's say in the C3 and the C4 pathway, C3 and the C4 pathway, it will have huge number of stomata. Maybe about 5,000 to about 30,000 stomata will be present in the leaves per square centimeter. Whereas when it comes to the CAM pathway, in the CAM plants will have maybe about 100 to about 800 stomata per centimeter square. So the number of stomatas have decreased. Not only that, in the CAM plants have got a very thick waxy covering around the leaves. So this also prevents or minimizes the diffusion of gases, loss of water molecules when carbon dioxide is taken inside. Another interesting aspect that is seen in the CAM plants is that in the internal structure where photosynthesis occurs, they are not close to the periphery of the leaves. They are about 7 to 9 cells away from the periphery of the leaves. So all these contribute towards ensuring that when the stomatas are opening in order to take up carbon dioxide, water is not lost or minimum water is lost. These are some of the adaptations the CAM plants have. Now we are going to look at how the CAM plants are going to carry out carbon fixation. We begin in the pathway starting with phosphoenol pyruvate. Phosphoenol pyruvate is present in the cytoplasm of the plants. To phosphoenol pyruvate, a carbon dioxide is added. A bicarbonate is added here. And to this 3-carbon compound, when you add another carbon atom, this is getting converted to oxaloacetic acid. So, in the oxaloacetic acid, which is a 4-carbon compound, that is how fixation of carbon is occurring. All this fixation of carbon will take place when the temperature is very low, when it is dark, and when there is more of water vapor available. So all this is happening in the dark. The conversion of PEP to oxaloacetic acid is carried out by an enzyme known as PEP carboxylase. This is the enzyme. This is one of the crucial enzymes that helps in regulating the CAM pathway. And once oxaloacetic acid is formed, let us see what happens. Oxaloacetic acid is getting converted to malate. And conversion of oxaloacetic acid to malate requires an NADH molecule. So, catalyzed by the enzyme malate dehydrogenase enzyme, malate is formed, and the malate is being transferred to a typical component or typical cell organelle, especially that is found in the CAM plants, and that is in the vacuoles. All CAM plants will have a vacuole which is used for storing this malate as malic acid. So, this, if you consider this is the vacuole, and this malate is transferred into the vacuole and it is converted to malic acid, writing the structure of malic acid, CHOH, CH2 and COOH. It's converted to malic acid. Now, malate is transported to, this is the vacuole, it is transported to the vacuole with the help of ATP expenditure. ATP molecules are needed in order to shuttle the malate by using a transporter. It is transported to the vacuole. As you can look at, when you look at the structure of malate, there are 2 protons that can be ionized. So therefore there are 2 pKa values. One pKa is at 3.4, another pKa 2, this is 1, pKa 2 is at 5.1. So therefore, no wonder why if the plant in dark or early morning, this will appear to be sour in taste because of this high acidity. So in this way, carbon that has been fixed is stored inside the vacuoles when the temperature is low and when the humidity is high, so that the stomatas can open, carbon dioxide can be taken inside, it can be fixed, it can be stored. This malate that has been transported out by using a malate transporter, once the malate is in the cytoplasm, this malate is taken up by the chloroplast. Let me create a little space. Once this is clear, now we are getting into what happens during daytime. So during daytime, this malate which is released, now the malate is in the cytoplasm. This malate which is released into the cytoplasm is taken up by the next cell organelle, that is a chloroplast. So this malate will get into the chloroplast. So here is the malate. This is a 4-carbon compound. Now malate will be acted upon by an enzyme which will remove a CO2 molecule from this. And that enzyme is a malic enzyme. Malate is acted upon by malic enzyme in order to remove a carbon dioxide. And this carbon dioxide is needed to be fixed for C3 pathway so that 3-phosphoglycerate can be made. Now, this is catalyzed by malic enzyme. There are 3 types of enzymes which can remove a CO2 molecule from malate. Which are the 3 types of enzyme? The first one is an NAD dependent malic enzyme. The second is NADP plus dependent malic enzyme. And the third one is a phosphoenol pyruvate carboxykinase. So different plant species will use one of these enzymes in order to remove decarboxylate malate. Once the CO2 is removed, malate becomes pyruvate. Now what happens to the pyruvate? This pyruvate will be converted to phosphoenol pyruvate. Now to convert pyruvate into phosphoenol pyruvate, we need to break down 2 ATP equivalent. So if the 2 ATP equivalents, in other words, 1 ATP is broken down to AMP and this is becoming 2PI. Catalyzed by a typical enzyme, name of the enzyme is pyruvate phosphate dikinase. That is the enzyme will convert pyruvate into phosphoenol pyruvate. So once you have phosphoenol pyruvate, now this phosphoenol pyruvate will come out of the chloroplast. So the phosphoenol pyruvate is coming out of the chloroplast, phosphoenol pyruvate. Once it is in the cytoplasm, phosphoenol pyruvate will now be converted to 3-phosphoglycerate. Remember, it is in fact the reversal of the glycolysis that will create 3-phosphoglycerate. So the 3-phosphoglycerate that is produced will be transported back into the chloroplast. So 3-phosphoglycerate is transported back into the chloroplast, and now this 3-phosphoglyceric acid is being part of the C3 pathway. So in other words, this will enter into the Calvin cycle. This will enter into the Calvin cycle. Along with that, the CO2 will be joining and that will continue to produce the triose phosphate molecules. Let me put this one here little more with greater clarity. So we are here in the phosphoenol pyruvate, which is there in the chloroplast, is taken outside. Phosphoenol pyruvate, this is converted to 3-phosphoglycerate. It is taken back into the chloroplast. This is the chloroplast. 3-phosphoglycerate is back into the chloroplast. This is the cytoplasm, and the 3-phosphoglycerate will enter into the Calvin-Benson cycle. So the CO2 which is produced here will be added to be fixed here. That leads to formation of triose phosphates. That is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. As we have seen in the C3 pathway, 5 of those will be returning back in order to synthesize ribulose bisphosphate. 1 of those will be made use in order to synthesize starch. So this is how a plant, especially a Crassulucean family plants related to that, the pineapple, the cactus, the orchids, aloe vera, all these plants, how they are able to handle, how they are able to survive an arid environment where the availability of water is very less.